So welcome again. Because we record now, I just repeat some welcoming words. I'm Alex Ecandi from ETPN. This is almost the last session of the METRINO Academy. And without further ado, I will leave the floor to my dear colleague Clémence for some housekeeping slides and introducing the session today.
Hi again. So yes, it's my pleasure to introduce this session today. And so as you know, today we are welcoming Ute Resch-Genger, and I will just, before she can start, I will just give you just a few words on the agenda. I will introduce very fast the METRINO Academy, and then we'll move on to Ute's talk. And of course, at the end, there will be the live Q&A during which you can ask all of your questions. Just a few housekeeping rules so that everything goes very smoothly. Your cameras, you can have them on. We are really inviting you to have them on if you want. And as you might have seen, your microphones are off and the chat should be disabled. It's just so you can focus really onto the presentation. And if you do have questions, feel free to use the interactive Q&A tool. It's at the bottom, you know, like the 3 dots on the bottom bar of Zoom, and you can open here a Q&A tool and type your questions. So just a few words very rapidly on the concept of the academy. Why are we doing an academy? First, we think that there is this need for pedagogy in nanomedicine, so we are covering one specific challenge for each session, and we designed them to be very actionable, to provide you with expert insights that you can actually reuse straight away in your lab, in your work. And we've designed it to be a monthly rendezvous and create this community. Very rapidly, a few words about who we are. What is METRINO? So METRINO is short for Metrology for Innovative Nanotherapeutics. And the aim of the project is really to democratize metrology standards in nanomedicine. It is big consortiums with university metrology institutes, and one SME and the ETPN Association. Who we are, so these are the pictures of all the work package leaders, and you probably recognize some of them because they were in the past sessions. So you can recognize Enrica from LNE, Robin, the project coordinator, Christian from PTB as well, Leonardo from INRIM in Italy, and today, of course, Ute from BAM. So the 3 pillars of METRINO, what we are doing is in the project is really to provide the community with reference materials, with SOPs, and also to have this community building action. If you are interested, we have this website, and because the project is coming to the end, we will publish more and more, and all the let's say the freshest results of METRINO will be available. So I really invite you to visit it. If you missed the past sessions of the Academy, we've had already 4 sessions. We can recognize some people. We know some of you have attended them already, but if you haven't, we really encourage you to just check the replay and the replay articles that we made. So a few words about who you are today. So mainly people representing academic institutions, but also metrology institutes, industry. And today the audience is quite diverse. We have people from over 15 countries, mainly from Europe, but also some people from Asia, from North America. So that's great to see. So I'm really happy to welcome Ute today. We are really lucky to have her. Surface chemistry is such a difficult topic, and it's really good to welcome Ute. And just a few words. So Ute heads the Biophotonics Division at the Federal Institute for Material Research and Testing, BAM. And her work focuses on optical measurements, nanometrology, and methods to quantify surface functionalities on nanomaterials. And she's also very involved in standardization activities in optical measurements and also nanomaterial characterization. So welcome, Ute.
So thank you very much for the nice introduction. So now it's time for me to share my slides.
Perfect.
So I will give you an idea about surface chemistry with respect to nanomedicine, and I will also provide you with some introduction to different methods and also some experimental data, so which I sometimes will just quickly go through. So why surface chemistry and why quantifying functional groups and coatings on nanomaterials? So here's the question is, what is your background?
Absolutely, exactly. I will launch the first questionnaire. It's just about knowing a little bit who's around the table today, and more generally regarding today's topic. So let's talk about surface characterization. And you, which type of nanosystem do you mainly characterize in your work today? Is it about LNP or lipid-based carriers in general, polymeric nanoparticles, inorganic nanoparticles, EVs? I work across several types, or I'm not directly involved in characterization. That's question number one. Question number two, what is your current approach to nanoparticle surface characterization? We routinely quantify functional groups or ligands. We characterize surfaces, but mostly qualitative way. We outsource this type of analysis. We do not characterize surfaces, or we honestly do not characterize surfaces, and we don't know how to do that. Which is perfectly fine. Welcome. This is a place to be today. And last but not least, what is your biggest challenge when quantifying surface functionalities or coatings? Is it about choosing the method? Is it about understanding what exactly is being measured? Is it about sample prep or workflow robustness? Is it about comparing results across methods, lack of validated protocols, limited resources? Would it be time, equipment, or you simply don't know or you don't need to characterize surfaces? So I will leave a few extra seconds so you can really answer the three questions, please. Let's have a look now at the answers. It's quite evenly, we don't have EVs people. They weren't interested, which is quite interesting, by the way, because EV surfaces are so complex, by the way. So lots of people on LNPs, polymeric, inorganic, also people working across these. So there's no big trend, I would say. Question number two is even more interesting because most of the people that characterize surfaces, okay, but in a qualitative way, not very in a quantitative way, but also some people that they do not know how to do that. And last but not least, methods, choosing the right method and really choosing what should be the measure and what should be measured is still the key challenge in surface characterization. So I don't know. I'm sure you would have tons of comments right on this first poll, Ute, but at least you know exactly what is the deal to convince people what is the right method, why they should do it, and which kind of knowledge is available to guide them through this surface characterization pathway.
So that basically is a perfect introduction for the lecture. So why surface chemistry and why is it relevant for coatings on nanomaterials and nanomedicine? So think about what surface chemistry does. It controls the dispersibility, so whether the material is hydrophobic or hydrophilic, the charge stability, so the agglomeration behavior, the processability. So if you want to bind a targeting ligand to the surface, sometimes function for quantum dots, and the interaction with the biological environment. Also, when you think about nanomedicine and the biological species, it's about biocompatibility, circulation time in the blood, so biodistribution, excretion, and binding to the target or unselective binding. So for targeted nanomedicines like liposomes, quantum dots, polymeric systems with antibodies on the surface, for instance, you have to bind the molecular recognition moiety to surface functionalities on the nanomaterial. So you also need to know what is there and how much. And then eventually which conjugation chemistry to use, which should be bioorthogonal to the functional group on the nanomaterial. So what kind of methods are there? So I took this overview from a recent review from Labuda. This gives you an idea about how to characterize nanomaterials. So you want to know something about shape, size distribution, composition, concentration, surface area, porosity, dispersibility, surface charge, and such parameters, but there's still relatively little standardized with respect to quantifying surface functionalities. So this was the reason why we got interested in this topic. Also have a background in quantum dots, and for quantum dots, surface chemistry is directly correlated to fluorescence properties. So you don't have standardized methods there and no reference materials with known functional group densities. So this was our entrance to the field a couple of years ago. And when you have a look now at the slide, it gives you an overview of the methods which we typically use in my lab. So with a green cycle around, we have qNMR, which is SI traceable. I will show you a bit later how this works. Electrochemical titration can be made also traceable, like conductometry, for instance. Then you have methods like XPS or TGA, IR spectroscopy, and a lot of optical methods measuring absorption fluorescence spectra. Since we also do a lot of photoluminescence studies, naturally, these are the methods which we have at hand anyway. What is important now with respect to the choice of a method should be suited for routine analysis. So it should be fast, easy, inexpensive, and if you have lots of samples, you want to automate it. Then you have to keep in mind what you want to measure. Are you interested in the total number of functional groups, which are relevant, for instance, for particle charge, zeta potential, or in the number or the amount of accessible functional groups? So this is a number which you need to know when you want to bind biomolecules to the particle surface. When you take a look on the scheme on the other side of the slide, you see there for labeling methods, providing you with accessible amount of functional groups, size of the reporter matters. So it's a difference between using small protons, which reach every functional groups, and larger reporters like certain dyes, or even large biomolecules. So this gives you an idea about why it matters what kind of, whether you have a labeling or label-free method, and reporter size can be relevant. So method validation implies also that you have to keep such things into account and use better multi-method approaches. So I will give you now an idea about the methods which we commonly use. Yeah.
I'm sorry, but am I right?
Yeah.
Should we do the second poll right now? I guess
okay,
so.
I'm sorry.
No, no problem. Can you go back on the last slide, please? It will be very quick. Okay.
Here we go.
Here we go. So the second one is more technical. I'm sorry. Yes, retour. Yep. It's about functional groups characterization. It's a quick question, but it's really to understand how you feel about that. So in your application, which information is most valuable? The total amount of functional groups, accessible amount of functional groups, both are important, or we are still figuring that out. The result is quite clear, I would say, is that both are important. How important are they to do what? I don't know the answers, but you do know the answer, Ute. So I'm sorry for interrupting, but we go back to listening.
No problem at all. So anyway, so let's move on. All right. Here we go. So these are the methods which I will show you today: optical assays, qNMR, titrations, XPS. These and functional groups which we normally study are amino groups, carboxylic acid groups. I don't touch thiol groups and azide functionalities, but they are also very relevant. So what kind of optical assays can you use and how do they work? So you can just go for a conventional dye bearing a reactive group, which you couple covalently to your functional group on the nanomaterial. And then you measure how much dye you have on the particle and maybe in solution. You can also go for chromogenic reporters. These are dyes which undergo a strong change in their optical properties upon interacting or upon reacting with a functional group on the nanoparticle surface. Thereby, the absorption fluorescence properties are changed. So you can distinguish where your reporter's been sitting. Then there's another type of probe, cleavable probes, taken from pharmaceutical industries or drug delivery systems. Here you have a cleavable linker, a reactive group, and a reporter group, and these probes can be detected on particle and in solution, and eventually adsorption, desorption assays, which rely on electrostatic interactions. So you have a negatively charged particle interacting with a positively charged dye and vice versa. So how does do such assays work? So when you have dye coupling to the nanoparticle, so you add your dye, then you label your particle with the dye, wash away the excess dye, and then measure the particle and the dye. So the problem here is you need to have several washing steps. Then the question is, do you have unspecific adsorption? And one is recommended here is often go for a mass balance. You know how much you add. Then measure the dye in the supernatant and measure the dye on particle. What you have to keep in mind when you do this, nanoparticles can scatter, so sometimes it's easier to measure the dyes which haven't reacted with functional groups on the particle surface. And then you have an indirect determination, which nevertheless works, but there you need to know how much dye you put in order to get this going. What can be also critical is how stable is your nanomaterial. So sometimes when you isolate your nanoparticle with a dye bound to its surface, you can just dissolve it and then measure the dye without having problems with interferences from scattering of the nanomaterial. This really depends on the optical properties of the nanomaterial, which you have to keep in mind. So these are the advantages and limitations of this type of assay, which is relatively easy. Alternatively, you can use a cleavable reporter. Such molecules are also commercially available. They are also used by industry. So here you have the same principle. You have your particle with the functional groups reacting with your probe. Then you cleave your probe and measure the probe in a solution after removal of the particle, so you don't have problems with scattering, absorption from the particle, and quantify your reporter. What can be interesting here is you can choose reporters which can be detected with different analytical methods like ICP, OES, XPS, if you have dyes containing heterofunctional groups like sulfur or nitrogen. In addition, you can always go for mass balances for method validation. So just to give you an idea about cleavable linkers, so these are systems which are taken from drug release concepts, so the hydrozones, pH cleavable. What we really like are the disulfide linkers. There are also commercial systems like SPDP available. You reductively cleave the disulfide bonds and you can quantify the sulfur on particle. You can quantify the probe in solution. And you see here, thereby you can do a straightforward method validation. So this is an example of how we do such a method validation. Here you see the so-called Almonds test, which is also taken from protein analysis. You can use this even for cell ligands and quantum dots, which we've published as well. So you see here the labeling steps, and you cleave off this product, which has a nice yellow color, which you can measure with a microtiter plate reader. And when you take a look on method validation, you see how much we look into how much amount was applied, then how much was cleaved off from the surface, and what do you find on surface with different methods. So photometrically, and with ICP OERs. And thereby, if this fits within the respective uncertainties, you have your method straightforwardly validated. Then there are other ways of using functional group assays. So a very common assay is a so-called Fluram assay, where you have this dye, which is non-colored, non-emissive, and this dye selectively reacts with primary amino functionalities, forming an absorbing and fluorescent products. So it was eventually first developed for protein analysis. So a lot of these assays have been developed for measuring functional groups on proteins. That's the reason why they're available for amino groups, thiol groups, or carboxylic acid groups. So here, when you take a look on the absorption spectrum, so the red spectrum is the spectrum of the Fluram, the dye. And when it reacts with amino functionalities, it forms a yellow product, and the emission also starts. The emission has a yellow-greenish touch, and it's quite intense. So you can use this. We used it first for micellar structures and quantum dots, but you can use it for all kinds of nanoparticles. So we used it, for instance, for aminated silica particles, for upconversion particles, bearing a silica shell with amine functionalities, or iron oxide particles with a silica shell and amine functionalities on the shell. The reason why we use it for these different nanoparticles is we simply wanted to see whether you have interferences caused by the optical properties of the respective nanomaterial. The nice thing is you can use it for a lot of different nanomaterials, and it can be automated, which we did. Because thereby you can screen a lot of different particles. And the reason why it can be easily automated is because you detect a product which is bound to the nanoparticle surface. So you don't have washing steps, which make automation of more complicated. But this assay is error-prone, so you have pipetting dilution steps, stability issues, so the product which you detect is not stable for a long period of time, maybe for 3 hours. And this implies that you really have to have a standardized workflow or protocol for the assay. So this is how we did this automation, very simple, with a pipetting robot and a microteeter plate. So thereby we developed a protocol. We eventually published this, and we use primary amine, ethanolamine, for calibration, because when you do an optical assay, you need to calibrate the fluorescence intensity measurements. So then we did, there was a fun context in the lab. So us, the group, 11 scientists, against the pipetting robot, the automation system, which I show you here. And we looked into how fast was a pipetting robot automation compared to what we achieved manually, with people who had a really good training. I even joined, and I don't do assays. I haven't done it for maybe a decade or maybe even longer. So everybody was allowed. When you look into the coefficient of determination calibration curves, the robot was better than the humans. Also, some experts were as good as a robot, but the robot can do this on a 24-hour basis. And for a human, this is a kind of exhausting. So it's possible to use simple automation tools for setting up automated assays. Another method, which I will only briefly touch, is pH titration. This is also something which you can do with an indicator or the pH electrode. It's the measurement is here the amount of deprotonable species. So you have ultra small reporters, and it can be automated with a titration setup. It is suitable for many nanoparticles, and it can be adapted to other functional groups. Redox titration, for instance, a requirement are nanomaterial must be stable at the pH values used, And you need to make sure that you can remove impurities like surfactants, excess coordinative rebound, surface ligand, something like this, because they also often contain deprotonable functional groups, which would otherwise mess up your assay results. So this is how you do this. And we decided to go for back titration. The reason for this was when you do optical measurements and you have a scattering nanoparticle dispersion, this makes it really difficult to detect a pH change with the eye or even with a smartphone camera. Think about 50 nanometer sized silica particles show up as a milky solution. So even with a strongly colored indicator dye, you don't have a chance. And they can also penetrate a pH electrode and mess up your electrode. So here you have the incubation of the nanoparticle, for instance, for the aminated groups with HCl, then you remove the particles and you do a back titration of your HCl solution with NOH and then determine the volume needed for these titrations. You know the mass of the particles and you need the surface area, and here you are. So this is a relatively easy way of determining functional groups like amino or carboxylic acid groups, but you can also use NMR. For quantum dots, you can do measured ligands directly by NMR. For other nanoparticles, it's a bit more complicated, but it's possible. So NMR is something which every chemist knows from preparative work. So you can do it for determining the identity of your product, purity, whatever. It's non-invasive, very chemoselective, provides you structural information, direct quantification, you count spins, so it's SI traceable, you don't need a specific standard, but it has less sensitivity compared to fluorescence, for instance. So what is important when you go do NMR? So you can do qualitative NMR as you're mostly familiar with, but you can do it in a quantitative way. Then you look into integrals. You need a standard where you know how many protons are relevant for which integral. You can do it with proton NMR, but naturally also with other types of NMR. And this is a method used for purity analysis in pharmaceutical industry nowadays, but it can be also used for ligand analysis. We used it, for instance, for silica particles. In this case, we had to dissolve the particles because otherwise, well, you don't see a signal on of the ligands on the particles. The particles are too large. You have too broad signals. They don't show up. So here I showed the signals we are looking into when you have aminated particles. We used malic acid, showing signals at different NMR fields compared to what you wanted to quantify. So what we did here first, we had our particle dispersion. We removed the particles by centrifugation. Then we dried the particles at elevated temperature in an oven, weighted the particles, dissolved the particles in sodium hydroxide, deuterated solvents, naturally added the internal standard ultra-pure malic acid for quantification, did the NMR measurements following a refined protocols, a number of scans, pulse angle, pulse delay. You had to make sure that your pulses are really properly go back to zero, spectral where such things matters. Then you do data processing, consider your satellites. We skipped the signal at 1.3 nanometers because particles were dispersed in ethanol before. So we wanted to make sure that we don't have interferences from ethanol. And we set up this protocol for 25, 50, and 100 nanometer sized particles because sure, particle size can, for instance, be relevant for centrifugation. So which speed is needed, such things. And this was refined with the aid of biocomparison with another group because we really wanted to make sure that everything works out and that sources of uncertainty have been properly considered. And the last method I'm going to show you is XPS. This is a very common method for surface analysis. Here you measure element ratios in the near surface region. Sorry for the typo. You can measure surface spectra and high resolution spectra. You can do it in a qualitative way, so which elements are present in the near surface region, which has a depth of about 5 nanometers. You can do it in a quantitative way. Then you have this ratio. You get information on the binding state of your element, the valence oxidation state, and it's suitable for all elements except hydrogen and helium. So it can be also performed with labels, text to increase the sensitivity. There you use often heteroatoms like fluorine, for example. So limitations are the limited information depths. Then when you use X-ray, well, you can have beam damage, so it's a destructive method. And the surface analysis of nanomaterials, there's a difference to 2D substrates films, requires simulation. You have to consider the size-dependent surface curvature, but this has been also established. So what is behind XPS? So this is basically Einstein. It's a simultaneous measurement of kinetic energy and number of electrons. So here you see the formula behind it, photoionization, the different binding energies of different carbon species, for example, to give you an idea what you also can learn from XPS. And it's broadly used. So, and what was now relevant for us was these 4 methods to do really a comparison with another group using different silica particles from commercial sources, varying in size, varying in preparation method, varying in morphology, amount of amine functionalities, just to get an idea. And this is one of the key messages of this presentation. So what happens when you use different methods with different limitations, providing different measurements, and why it's so relevant to do multi-method studies, and also why it is relevant to do it together with another group, because another group is not as biased as one group, because you discuss protocols, measurements with your colleagues, so thereby you're automatically biased. And we wanted to use this study to develop and optimize protocols for a broadly run international comparison. So when you take a look now into the data here in the silica particles comparing the Fluram assay and the titration, you naturally see the numbers differ. They must differ because you use protons, which are ultra small, grab every functional group on the surface, every amine functionality, whereas the Fluram assay cannot bind to every functional group. The dye is too large. So this gives you an idea about why it's important to have an idea about, do you measure total or a number of derivatizable conjugatable groups? Then when you compare the titration method with qNMR was done by 2 labs. So you see if 2 labs, 2 experts do such studies, the uncertainties are relatively small. The data are supposed to match. And the NMR data match with the pH titration methods. So this also thereby we validated our pH titration. Then we compared XPS and the other methods. And one of the reasons why I show you this is when you look now into the particles, we had mesoporous and porous particles. For the porous particles, we had a relatively good correlation between XPS and qNMR. But for the mesoporous particles, well, it didn't work out. The reason for this is also quite natural. For NMR, you dissolve the particles. Well, you grab every functional group which is there. For protons, they are small enough to penetrate the pores. The dyes, maybe one dye has a chance, but that's it. But for XPS, you're limited with your penetration depths. So automatically you're supposed to find much less functional groups on the particle compared to qNMR in solution, for example. But this can be also used, for instance, estimating monolayers on particles, getting an idea of indirectly surface morphology. So multi-method studies can help you to learn a lot about your particle. So what is the take-home message now? So criteria for method selection, signal generation principle. Think about what's behind your method. Is it a labeling method? How do you generate your signal with a reporter? Which size? Which measurand is quantified? How chemoselective is your method? What could be possible interferences from other compounds may be present in solution? Then method and material-specific requirements. So for instance, something like size, optical properties of materials, requirements on calibration, sensitivity, limit of detection, which accuracy do you want in general, because this automatically increases the more accurate you want to be, the more amount of the higher the effort which you have to dedicate to calibration and refining protocols. Sample preparation protocols, workflows are relevant because this is often a major source of uncertainty. Then speed of analysis, analysis time, how many samples as well, cost, instrumentation, manpower. Maybe you're fine with very simple methods, validate them once by comparing them with a more advanced, sophisticated method, then go for your simple method. Then can you automate it? What can you buy with respect to automation tools? And for some requirements, ISO EN 17025, that's about traceability of your method. Validation showed you how to do this with the aid of multi-method studies, validation via cross-comparison. So what is then eventually, what can you do with such things, or why do we do this? So we have a standardization project behind it. So we want to develop methods for characterizing, quantifying functional groups on nanomaterials and also on coatings. So this was approved in 2022. And we recently filed for a new work item proposal. So this is basically then the international standard. And what is behind the standard? Well, this will be the protocols which we jointly developed in this bilateral comparison. Then we refined this in a comparison in this consortium SMURF Nano with different partners from industry, from National Metrology Institute. And now we are running a huge interlaboratory comparison under the roof of WAMAS with groups from academia, industry, and under CCQM. This is what is behind the National Metrology Institute. This study will then be used for setting up standards for qNMR and XPS. And with this, I thank you for your attention. But now we have another poll.
Yes, we do. Fantastic job, Ute. I always say that, but it's true. I've learned a lot. And that was an extraordinary summary of the field. And I mean, you just follow the last slide and you will do a better job in characterization surface chemistry groups. We will start the Q&A in a few seconds, but before that, I strongly encourage you to have a look at the Q&A tool. You just click on the plus on the toolbar of Zoom at the bottom right, the three dots, more or plus, and you see the Q&A. This is the only place to ask Ute a question, but maybe first some key comments from Clémence.
Okay, good. Thanks, Alex. Yes, so we really encourage you to engage now with Ute. So as Alex said, you can type your questions in the Q&A tool. You can also vote for the questions you like most. So go for it. Just a few words as well. So we right after the webinar, you will receive a satisfaction survey It really takes 2 minutes to fill, so it's super. I mean, it's really important for us. It's really helpful for us to to keep improving our webinars and events. So please take the time to answer the survey. We will also like to invite you to save the date for May 12, during which we will have the final session of the METRINO Academy, and it will be part of actually the final meeting of the METRINO Consortium, because as you might know, it is ending very soon. So we will share more information soon, but save the date already for May 12th. Okay, and finally, please scan the QR code for information on METRINO. You can also contact us. We are really happy to have you getting in touch with us. And now I'll give the mic back to Alex.
Yes, I'm back with the last poll. I launch it right now, and please don't take it as an excuse not to ask Ute's questions. She's waiting for you. How could surface characterization be easier for you guys? And what would help you most in your work today? Is it about a practical guidance on method selection, like Ute in a digital tool? Is it about better reference materials? Is it about getting access to more standardized workflows and reporting, easier to implement automated methods, better links between measurements and biological performance, or accessible, reliable, and well-documented existing data? It's quite well, I mean, you need all of that. That doesn't help, guys. We doesn't have the resources to help on all of these. But maybe, yes, before the end of the METRINO project, we will produce some reports, some deliverables, but also some guiding tools to try to bring our support specifically on that. I guess, Ute, there's among all the fields that we've talked about in the academy, this one is strong. I mean, the need for help is enormous here.
I mean, just one of the reasons why we basically publish all data which we obtain, also the protocols, because we think it's very important for the community to have access to this knowledge.
It is. It absolutely is. We'll start by the Q&A. Thank you very much, Jean-Michel Rabanel, for asking your first question. So detection and quantification are issues, but if you want to get data such as ligand densities or other figures are needed, numbers of particles, their diameters, this later figure usually is derived from ensemble measurements, hiding eventually subpopulation. How do you think we can tackle the possible ligand density heterogeneity in a particle population, if you think it's a real issue indeed?
What you can do is you need to know the particle size. Then you need to know the number of ligands per particle in average. And what you could also do is you can do some quantum chemical modeling of how much space a single ligand needs, and basically how many ligands could form a monolayer on your particle, depending on in which conformation they exist, whether it's extended or banded or whatever. So thereby you at least get average numbers. So this is what we did, for instance, for quantum dots.
I don't know if that's the hidden question, but do you think we should care about that more? I mean, is it? Do you think that we have a strong functional need about the functionality, for instance, of grafting some targeting molecules, or like the density and the heterogeneity is really a functional issues, or we should? I mean, we should study it, but we should not be too worried about that. Do you have any vision on that?
Are you asking me or?
I'm totally asking you to.
Yeah. Okay, I just wanted to make sure. Well, there are a lot of papers describing how important it is to have directional binding of antibodies. So you really need to make it's even more complicated. So what you ideally want is a very monodispersed particle size. Then you want to control the number of ligands per particle, and ideally the number of targeting or biological recognition moieties per particle, because there are reports on that the number can also affect the binding constant, for example. And now you can imagine the smaller the number of particles as you're looking at with your method, the more heterogeneity concerning particle size, ligand density distribution, number of biomolecule ligands on particle surface matters. And there you also need to distinguish between, do you know the number of biological recognition moieties, or are you interested in the number of functional biological recognition moieties? Because when you do, for instance, a protein assay, well, you measure protein concentration, but this doesn't tell you whether all proteins which are there are really fully functional. So there are lots of papers on such things or on different types of nanoparticles on gold. I'm aware of only one paper on gold where people managed to eventually have gold with a single antibody bound to its surface and showed how important this was for the binding constant. But these are the general challenges. So you can imagine this basically for quantum dots, this led to a decade of ligand design, how to do it best, how to make sure that the ligand remains on particle and doesn't desorb because it's coordinatively bound, then that all the binding moieties are really directed in the right way, such things. But this implies a lot of method, and binding constants of antibodies on particle to the target are really difficult to obtain. You can do it with a functional assay, or you can do something like ITC, or you can use the biocore system, for instance. But this is something which is really challenging. The methods are there, but it's challenging.
And costly, I guess. So you need to really decipher whether or not you need that information. Maybe the next, sorry.
What you could do is you could use, if you have a biologist at hand, you can do it with a cell assay, which could save you a lot of money.
You need two things. You need a robot and a biologist.
Yeah.
Yeah, okay, that's a good conclusion. Next question. Let's talk a little bit maybe about the uptake of these methods, or at least this attitude. So maybe Clémence, you can go for the next one.
Thanks. Yes, so just some practical update question. Some wondering for a lab or a company that does no surface chemistry. I mean, they know it's important, but they don't have a workflow yet. What would you recommend them to do first? Where should they start if they were to implement, very robust workflow?
I mean, optical assays are normally a good choice because a lot of groups have microplate readers at hand or spectrometers, and there you have the protein assays, which have been tested. They are on the market for 20 years, something like this. So this is relatively easy. I mean, NMR is great because at universities you normally have NMR facilities, and also having NMR measurements done is also not utterly expensive. So there you need to think about, is your particle stable enough? Can you dissolve it? Such things. So this could be also a nice method. XPS is something which is costly. There you need the instrument, but also the XPS expert. Titrations are nice as well. There are also lots of titration methods, redox methods as well. So sometimes the good old methods are not the a bad option to go for if you want to spend or get started and do not have a lot of money at hand.
Okay, that's great. And also you've just showed in your presentation that you get consistent results, whether you were doing qNMR or the titration. That's amazing. So another question. So you gave a practical set of criteria for method selection. Which one do you think people tend to underestimate the most? Could it be, I don't know, the cost or the measure on the chemoselectivity?
Well, in my opinion, the sample preparation workflow, because, for instance, for the qNMR workflow was about we had higher uncertainties for the smaller particles. This was simply related to difficulties in centrifugating or removing the particles, the 25 nanometer sized particles, from dispersion. And at first we thought, well, maybe a size effect, right? But it was just such a simple issue, or how to make sure that your particles are properly dried or fully dissolved or do not stick to whatever. It's also a major source in uncertainty for XPS, for example. For optical methods, it's generally scattering, which can cause problems, unspecific adsorption.
Okay, so, sorry. So what would be your best advice for people to improve their sample prep?
Well, normally it's good to try it with two people because it's unlikely that two scientists do the same mistake. So ask your colleagues, you have your protocol, give it to your colleague with the same particle batch and figure out whether you get matching data. I think that's the easiest way. Or if you have a person from a different group using a different method, and then you can use this for comparison and think about setting up reference materials. You can basically characterize your own samples. If you know that they're stable enough, you can use them for setting up methods in other labs.
I have several questions for you, Ute, but please, the others, don't be shy once again. Even the people from Metrino can ask questions. Yeah, so it's crystal clear what you said about the academic labs that can access pH titration. Normally you should do, but also you have probably NMR in the university. But if you're looking for more industrialized process, what would be the methods that are a bit more costly, but that you would recommend to do on a more systematic point of view when money is a bit less a challenge and you really want to do something that is ready for translation to the clinic? What would be the advice that you would give not to an academic group, but to an SME or a larger company?
I mean, I know that a lot of SMEs also use the optical assays. By the way, we started with functional group analysis because a company wanted us to develop workflows for them. So this was the reason why we started with the assays. This is what they were using for stability, production control, and aging monitoring. So, and normally you'd use such methods which you can easily run on a large scale, validate them once with NMR XPS. And for larger companies, some companies also use solid-state NMR. For instance, when you work with silica, solid-state NMR is a typical method. Sometimes you could think about HPLC, could be also used. We have papers on HPLC as well. But I can really recommend the titration methods. I know that they're broadly used in industry and also in larger companies.
Great. I ask this question at every speaker of the METRINO Academy because I think it's important to talk about that for 2 seconds. Okay, so you show us what to do. Can you please show exactly what stop doing? What is the worst that you see every time? Like people think they do nice characterization of surface, but it's really not enough, or it doesn't tell exactly what they think they are telling, or it's not really comparable. So what is your top 1 or top 3 things that you would like us to stop doing in the lab?
Well, I think with respect to optical methods, avoid scattering. And think about, I mean, I showed you how you can avoid this with the right choice of an assay. If you have particles which are strongly colored or strongly scattering, then you're better off with cleavable probes or measure particles via a mass balance and remove the particles first, because otherwise you always have this background which could cause problems. With the Fluram assay, we observed problems. In our case, we always got it going, but we know from others it's somehow tricky. If you know that you try something which apparently doesn't work very well, maybe don't spend too much time on it, ask somebody else to repeat it. If this person is also not capable, skip it, go for another method. Don't waste your time on something which, for whatever reasons, doesn't work very well in your lab environment.
That is crystal clear, I guess. Maybe we should move towards the end of the webinar, but we have some questions more about, let's say, the community and the impact for people. Oh no, I've just forgot a question I want to ask you, Ute. Oh, I was amazed about the reproducibility. I don't know if it's the right word between the two labs. I mean, it's really impressive. But okay, you're a metrology institute. I mean, hopefully you can measure things. But do you think that for people on the nanomedicine side, like technology developers, is it easy, do you think, to build a scientific collaboration with people just on surface characterization that say, we want to publish, but we want to show credible results? Would you like to compare our particle with that? Would you encourage to do a collaboration just on this specific part? Or you think it's a bit tricky for a nanomedicine lab to ask that to colleagues?
To be honest, I have great trust in industry. So we have a lot of collaborations with industry, and normally they have well-established protocols. I really work on them. We refine them with them together, and then you achieve uncertainties in the order of 10%. So this is feasible.
On bad days.
Yeah. With respect to NMR, think about how long NMR is on the market. So how much experience people have with NMR. It's much more like in the quantum dot community, you nowadays use NMR for ligand analysis because it tells you quite a lot. It's maybe less common for silica materials, but if you think about dissolving particles, you can measure NMR spectra very accurately. So there, it's if you get the sample preparation workflow running. I mean, we got the NMR measurements were done at a university. We are currently comparing results from an ILC from universities who've often never worked with a nanoparticle before. They were trained based upon our protocols. So we spend a lot of time on developing the protocol. They do very well. For them, it's an NMR expert. So our measurements were run at Free University Berlin. And for them, it's like, why is it so interesting for you? For us, it's boring. So they do they do research on NMR. For them, it's like easy peasy. It's so this is just a completely different point of view. Think about people doing protein NMR. All the kinds, dozy, nosy, whatever, functional, fancy techniques. For them, this is easy work. Quantifying with NMR. Yeah.
For them. Okay.
Yeah.
So you strongly encourage to ask for support and help and collabs. I mean, what might be difficult for a lab is like normal life for another lab, right?
Yeah. But you pay maybe 50 euro for an NMR spectrum from a university, and that's it. It's not expensive. That's the reason why I'm suggesting this. So set up your own methods in industry in the lab, your assays, whatever. Do NMR once in a while, pay a university, and here we are. And it's a very efficient way.
Fantastic, crystal clear. I mean, the plan, but also the delivery of the plan. I guess, Clémence, you have a question more on the impact for the community.
Yes. So you've mentioned, validated workflows, ILCs, standards like the one you've submitted. What do you think is the biggest priority for the community in a very short-term way?
So I think there it's about figuring out which uncertainties you can tolerate, because this determines the amount of work which is required for setting up or harmonizing methods. So this is what I'm also telling everybody with respect to luminescence techniques. So if you can live with an uncertainty of 50%, maybe you don't need a calibration or a calibration once in a while. If you want an accuracy of 1%, it's a different story. So really think about what you need. Or do you simply want to look into changes? Then it's also a different way of, or this automatically implies less importance with respect to method development. Then it's about how robust is your method, how well is the reproducibility. But when you simply are happy with looking into changes or comparing things, this could save you a lot of time. So really think through what do you want to achieve, because this could be expensive. And if you know what you want, this could save you a lot of time.
I guess also the second question behind Clément's question is what is the difference for the community of having ILC that are published and you can use them and you see the interest? You see that sometimes pH titration is equal to an NMR. Maybe you don't need NMR. And also you see sometimes that it's not that easy to compare with cross laboratories. So what is the difference for you that you build standards? What is the difference of having a standard available being compared with just ILC published? What is the real added value of a standard? I just want you to share your experience because you're making standards.
Yeah. So basically, the ILC is, in my opinion, the most important step, because this is also about group bias. You need several groups with different backgrounds to really figure out where could be problems with respect to a certain procedure, with respect to a certain protocol. Because if you develop this, you automatically do it correctly and you communicate it within your group. But if somebody else does it and has a different background, maybe they simply don't know something or do something automatically different because they have a different knowledge. So, and this is a nice thing about, or that's a reason why ILCs often come first before you have a standard. You want to see how well do groups do with a different background, different instrumentation, different operators, with respect to comparability and certainty, because only then you can be sure that a method has a certain uncertainty and is robust. And in nanomedicine, this is even more important, because there it's, I mean, when you measure all kinds of health-related biomarkers, whatever, you want to make sure that the data are comparable. So there's sufficient training about making methods robust, harmonized, whatever. And ILCs are, in my opinion, the only way of doing this.
Sorry for the silly question, but it's a genuine one. Is that easy to create an ILC when you want to develop your nanomedicine with a marker that is you want to characterize? For instance, would you take some collaboration with people interested in doing better surface and doing ILC with you?
Sure.
Or okay.
I mean, we're very open. So basically, we have this European metrology project, and we started approaching university groups. There were some university groups, really NMR people, very excited about joining the ILC. One NMR group recently offered us, they have a protocol, it's a French NMR group, for automated data evaluation of NMR spectra. So we were very happy. This is what we're currently evaluating. Then we're going to use this for the ILC, validate this procedure as well. So I think this is a bit about open science. So we will publish the protocols from the ILCs together with all participants. And most likely, if everybody agrees on it, we will also upload the data on Zenodo.
That's fantastic. I mean, This is what we need. And this is exactly the purpose of these kind of metrology projects, such as METRINO or Smart Nano, is not just to create knowledge, but to share it in a very open way. So I truly appreciate. We take a last question from the crowd. This is from my dear colleague, Speranta. Hello, Speranta Tănăsescu. Okay, two questions, Tristan. The first one is, I formulate again. So thank you for the presentation. We are currently with the isothermal titration calorimetry, and the thermodynamic parameters could be valuable descriptors of the interaction at bio-nano interface. We usually compare with nano-DSC results, giving information about the proteins, for instance, stability. Do you think to be used more in the future for the metrological community information?
So we've never done nano-ITC. I know it's a very challenging method because you measure very small numbers. And so I really cannot tell you much about it. I know it's a very important method. You need these interactions.
Okay, if you want to try it, Speranta is the best expert that I know, so you should try with her.
But this is really good to know because we could be interested in
I can put you in contact quite easily, and let's see what happens.
We've never done this because this is not our area of expertise. We know that it's a very challenging method, and we really like to collaborate with people because I really like to learn from others as well.
So this is the easy peasy part is for Speranta, this part. Okay, next one. Thanks again for the nice question, Speranta. Tristan, who I must say got an award among the organizers of the METRINO Academy of the most trustable attendee there every time, one of the first to register. Tristan, we truly appreciate that. You can hear that from ETPN side. So is the method panel as various for investigating surface of organic polymer NPs as of inorganic NPs?
So we have experience with polymer particles, polystyrene, PMMA. So this is which has been studied by us as well, quantum dot silica nanoparticles. We don't have much expertise with lipid nanoparticles. We once studied quantum dots with a lipid shell. And there the methods worked out. For instance, this is where we first started with the Fluram assay. There we even learned something about which functional groups were sticking to the outside and which were bound to the particle surface in this bilayer structure. This is all what I can tell you with respect to lipid nanoparticles. We know from groups which approached us that titration methods are used for liposomes as well. So for carboxylic acid groups, for amine functionalities.
Okay, I hope it answered the question, but by seeing Tristan's nodding, I guess so. Last but not least, Brinduza, another very trustable attendee, almost there every time. Good afternoon, Brinduza. You said, unfortunately, I could not join at the beginning, so maybe the answer is already provided. How can nanoparticles be designed to escape the immune system while simultaneously respond to dynamic and rapidly fluctuating pH condition? Can this be reconciled? Thank you. Do you have an answer on that, or a view on that?
To be honest, I don't understand the question. So could you maybe explain?
No. I'll do something that we don't usually do during these events. I leave the mic to Brinduza. Brinduza, you can reactivate your microphone right now and directly open your camera and ask your question, or maybe make it more precise for
would be best,
Hi, everybody.
Hi.
Thank you. Unfortunately, I couldn't join the meeting at the beginning. Perhaps my question was not very clear because Alexandra said, okay, we are at the end. Actually, when you design nanoparticles with some functional groups on the surface, we want to be sure that the immune system does not remove them from the body. But in the same time, we have to decorate the nanoparticles so that they can release the drugs depending on the pH. And I'm wondering if these two are how to say, I cannot find the word now.
No, I understand.
I think that you got my point.
I mean, to be honest, I lack the biological knowledge. So what you could think about, but this is something which you most likely know better than I, is whether you go for a different release mechanism, maybe not pH cleavable linkers or something like this, go for something which react to reductive conditions or oxidative conditions, because there are many other ways of having such a mechanism started. So this is the only answer I could give you, but this is really not my area of expertise.
My question was limited to pH, but in reality, the conditions are more complex, and there's a lot of stimuli that can come.
We have collaborations. We had a lot of collaborations with medical doctors and biologists where we did the spectroscopy, and they used nanoparticles with antibodies and then dyes which could be activated under certain pH conditions to become fluorescent. This worked. And they really tackled cancer cells. So, but in this case, it was Herceptin and particles overexpressing her cells overexpressing HER2/neu. So there you really had this nice mechanism, particle binding, being internalized and then becoming emissive within the cancer cell. So you could even fluorescently label the cancer cell. I don't know how well such things work with a drug, to be honest. This is something which we never tried.
Thank you. Because actually, we have to think from economical point of view. Let's say that you design a nanoparticle to respond the pH, so you put something on, but let's say the immune system will remove them. So you put it there for nothing, you know. You just lost resources and the time and everything.
So honestly, I don't know which models you have in order to test this in advance to make sure that the nanoparticles go where they're supposed to be going. I mean, I know that this really tackling a biomarker on a cell and hampering excretion and really making sure that circulation, everything fits well is a really huge challenge. So what I'm doing is easy peasy compared to this.
Well, you know, it's always complex. Thank you, Brinduza. It's always complex when you look at what others do. First of all, I'd like to express my deepest gratitude to you, Ute, for preparing such an impressive set of slides and knowledge available to in very pedagogic point of view. So first of all, bravo et merci beaucoup, Ute. Second, I would like to also thank. No, that's I'm seriously. I mean, I've never had such a deep conversation about surface. So even on a metaphysic point of view, that's quite unusual for me. Thanks also to Clémence that organized together with you this almost last session of the Academy. As you know, the replay will be available. Please take 30 seconds to answer the satisfaction survey. We take really into consideration and into actions what you said for the next and last event. And that's almost it. Ute, do you want to add something? Or did we say almost all of it?
I wanted to thank the audience for the active participation. I really enjoyed it.
Fantastic. We did enjoy also. Thank you all for attending until the end. And I really hope that you take some time to participate in our stakeholder engagement, because we need your expertise, not just to share what METRINO did, but to have an impact on your daily lab practice. So how do you say that? Stay tuned, exactly, and have a wonderful end of the afternoon. Bye-bye.
Bye-bye.
Bye. Thanks, Ute. Thanks, everyone.